Hook. Write in the comments where you're watching from and what time it is, so I can get to know you better. To support the channel for free, don't forget to subscribe and like. Your brain is smaller today than it was yesterday. Not by much, not in a way you can feel. But the process is happening right now as you watch this, and it has been happening every single day since you turned 25 years old. This is not a disease. It is not a disorder. It is one of the most consistent and well-documented biological processes in all of neuroscience. The human brain begins losing volume at a measurable rate in early adulthood, and in most people, that process accelerates quietly through their 30s, 40s, and 50s before producing effects they can actually notice. By the time most people realize something is different about their memory, their focus, or their processing speed, the structural changes driving those effects have been accumulating for decades. Here is what makes this genuinely important rather than simply alarming. The rate at which your brain shrinks is not fixed. It is not purely genetic. It is one of the most modifiable biological processes researchers have identified in the past 30 years. The same behaviors that accelerate it and the same behaviors that slow it almost to a standstill have now been studied extensively enough that the picture is clear. In this video, we are going to trace exactly what brain shrinkage is, why it happens, what accelerates it beyond the normal rate, what the early warning signs actually look like in real daily life, and what the research consistently shows can preserve brain volume in ways that were not considered possible a generation ago. By the end, you will understand something about your brain that most people never learn until it is significantly harder to address. Section 1. What brain shrinkage actually is. The science of volume loss. Let's begin with what is actually happening at the cellular level, because the term brain shrinkage sounds alarming in a way that obscures what the science actually describes. The brain is not a fixed structure. It is a living, dynamic organ that is continuously changing, forming new connections, pruning old ones, and responding to everything you experience, eat, stress over, and sleep through. What researchers measure when they talk about brain volume is the physical size of brain tissue as seen on MRI scans. And what those scans consistently show is that this volume decreases with age in a predictable, measurable pattern. The primary driver is not neuron death in the way most people imagine. The brain does lose neurons over time, but the more significant contributor to volume loss is something called dendritic pruning, the reduction of the branches and connections between neurons. Dendrites are the tree-like extensions that reach out from neurons to form synaptic connections with neighboring cells. When these connections are used regularly, they are maintained and strengthened. When they are not used, they are trimmed back in a process that the brain's immune cells, called microglia, carry out continuously. The result is that brain volume loss is not simply about neurons dying. It is about the network becoming less dense, less connected, and less elaborate over time. The regions of the brain most vulnerable to age-related volume loss are the prefrontal cortex, the region responsible for executive function, decision-making, working memory, and impulse control, and the hippocampus, the structure most critical for forming new memories and navigating spatial environments. These are not peripheral structures. They are the regions most central to what we think of as cognitive sharpness. What research has established over the past 2 decades is that the rate of loss in these structures varies enormously between individuals, and that the variation is far more strongly predicted by lifestyle factors than by genetics alone. Section 2. What accelerates it? The surprising drivers most people never consider. Here is where the science becomes genuinely counterintuitive, because the factors that most powerfully accelerate brain volume loss are not the obvious ones most people worry about. Chronic psychological stress is one of the most reliably documented accelerants of brain shrinkage, and the mechanism is specific. Cortisol, the primary stress hormone, when chronically elevated, is directly toxic to hippocampal neurons. The hippocampus contains a high density of cortisol receptors, which means it is exquisitely sensitive to stress hormone levels. Research consistently shows that people with chronically elevated cortisol have measurably smaller hippocampal volumes than age-matched people with normal cortisol levels. The effect is not subtle. In some studies, the difference is equivalent to several years of additional aging. Sleep deprivation is the second major accelerant, and arguably the most underappreciated. During sleep, the brain runs a waste clearance system called the glymphatic system, in which cerebrospinal fluid flows through the brain tissue and removes metabolic byproducts, including a protein called amyloid beta that accumulates between neurons. Amyloid beta is the same protein that forms the plaques characteristic of Alzheimer's disease. Even a single night of poor sleep produces a measurable increase in amyloid beta levels in the brain. Chronic sleep deprivation allows this accumulation to compound over months and years, creating a neuroinflammatory environment that accelerates structural deterioration. Physical inactivity is the third driver, and this one surprises most people because the connection between movement and brain volume is not immediately intuitive. Skeletal muscle, during contraction, releases a protein called BDNF, brain-derived neurotrophic factor, which is essentially a growth and maintenance signal for neurons. BDNF stimulates the growth of new neurons in the hippocampus, strengthens existing synaptic connections, and directly counteracts the pruning processes that reduce brain volume. Regular physical activity maintains chronically elevated BDNF levels. Its absence removes one of the brain's most powerful self-maintenance signals. Section 3. What it looks like in real life. The early warning signs before the big ones. One of the most frustrating aspects of brain volume loss is that its early manifestations are easily dismissed or misattributed, and by the time the effects become undeniable, significant structural change has already occurred. The earliest sign is almost universally a change in working memory, the ability to hold and manipulate information in your mind in real time. This is not the same as forgetting things from years ago. It is the experience of losing the thread of a sentence you were forming, forgetting why you walked into a room, struggling to track multiple pieces of information simultaneously in a way that used to feel effortless. Most people in their late 30s and 40s attribute this to stress or being busy. Sometimes that attribution is accurate, but when it is consistent, when it persists across different stress levels, and when it is accompanied by other changes, it is worth paying attention to as a signal rather than dismissing it as circumstantial. The second early sign is processing speed. The brain communicates through electrical signals traveling along myelinated nerve fibers, myelin being the fatty sheath that wraps around axons and dramatically increases signal transmission speed. When myelin integrity declines, as it does with age and particularly with chronic inflammation and nutritional deficiencies, signals travel more slowly. This is experienced as a subtle but real increase in the time it takes to respond to new information, to switch between tasks, or to find words during conversation. The pause before the name comes, the moment of searching for a word that used to arrive instantly. The third sign is spatial navigation difficulty. The hippocampus is deeply involved in building and maintaining internal maps of environments. Research has found that difficulty navigating familiar places or a reduced ability to form accurate mental maps of new environments correlates strongly with hippocampal volume reduction. None of these signs in isolation is diagnostic of anything serious, but their combination and their trajectory, whether they are stable or gradually worsening, provides meaningful information about what is happening in the underlying structure. Section 4. What the research consistently shows can preserve and even restore brain volume. Now let's address what the science actually supports, because this is where the conversation shifts from concerning to genuinely actionable. The single most robustly documented intervention for preserving hippocampal volume is aerobic exercise. This finding has been replicated across dozens of studies using MRI measurement, and it holds across age groups, fitness levels, and genders. A landmark study showed that older adults who completed a moderate aerobic exercise program over the course of a year showed measurable increases in hippocampal volume, increases that reversed roughly 2 years of typical age-related shrinkage. The mechanism is BDNF, the same growth factor discussed earlier. Exercise produces it in quantities that no other intervention has been shown to match. The dose required to produce measurable effects is not extreme. Research suggests that consistent moderate aerobic activity, accumulated over time, is sufficient to produce and sustain meaningful neurological benefits. Sleep optimization is the second intervention with strong structural evidence. 7 to 9 hours of consolidated sleep allows the glymphatic system to perform its clearance function completely. Reducing chronic amyloid beta accumulation reduces neuroinflammation, which is one of the primary drivers of accelerated structural decline. The research is consistent in finding that sleep quality, not just duration, matters. Fragmented sleep, even at adequate total hours, does not produce the same glymphatic clearance as deep, consolidated sleep. Nutritional support for brain structure has the most nuanced evidence base, but the clearest findings center on omega-3 fatty acids, specifically DHA, docosahexaenoic acid, which makes up a significant portion of the phospholipid membranes of brain cells. Research shows that adequate DHA intake is associated with greater brain volume, better myelin integrity, and reduced rate of age-related structural decline. Chronic DHA deficiency is associated with the opposite. Chronic stress management produces measurable effects on hippocampal volume, not through awareness or psychological reframing alone, but through interventions that produce actual physiological reductions in cortisol. Research on meditation, specifically the kind practiced consistently enough to produce measurable reductions in cortisol and inflammatory markers, shows correlations with preserved hippocampal volume that are not seen in control groups. The picture that emerges from 30 years of neuroimaging research is one where the brain's structural trajectory is far more plastic, far more responsive to daily inputs than the previous generation of neuroscience ever suggested. Your brain is not on a predetermined path of decline. It is on a path shaped continuously by what you ask of it, what you feed it, whether you move, and whether you rest. It is shrinking. But by how much and how fast is still, to a significant degree, up to you.